Hello friends, so next question is what is the number of hydrogen bonds in the double helical B DNA structure of 100 base pairs between 20 adenine and 10 thymine in one of the two strands? Okay, so here it is given there are total 100 base pairs. Okay, among the 100 base pairs they are 20 adenine and 10 thymine in one strand. So we know that adenine and thymine forms two hydrogen bonds, whereas the guanine and cytosine forms triple hydrogen bond. Okay, so now here 20 adenine, that means in the other strand it is going to be 20 thymine, it is going to be 20 thymine okay, they are 20 thymine, and if they are 10 thymine here, that means they are 10 guanine here. Okay, so total here 20 and 20 adenine, 20 thymine, 10 thymine, 10 guanine. That means we are having 30, 80, which are forming two double bond. We know A and T is forming two hydrogen bond. That means 2 into 30. Okay. Now this is about 80. Now let us see for G and C. G and C forms triple hydrogen bond. So they are 30, 80 among 100 base pairs. That means 70 are GC and GC are forming 3 hydrogen bonds. So we can do 3 into 70, which will be equal to 210. When we add 3 to the 16, that means 160. Oh sorry, 16. 6060. Okay. So yeah, here we are going to have 60. 210 plus 60 will be equals to 270. So there are total 270 hydrogen bonds formed here. So here correct option is the third option. Okay. Next question. Percentage of sequence identity for the following aligned sequence is. So here see, it is very easy to calculate the aligned sequence. Here we have to see how many are matching here, aligning with each other. So here we have 1, 2, 3, 4, 5, 6 and 7. So 70% is the correct option. Next, in the lag operon, promoter and operator refer to. So see, this is the lag operon. Okay. In the lag operon, we have promoter sequence and then we have operator sequence. So on the promoter sequence binds RNA polymerase, whereas on the operator sequence binds the repressor. So here correct sequence is binding site, correct answer is binding sites for RNA polymerase and repressor. Okay. Next question. PDB contains information about. So PDB stands for protein data base or protein database bank. Okay, so it contains the information about the tertiary structure. Next question. The primary RNA transcript of the chicken ovalbumin gene is 7700 nucleotide long. So it is 7700 nucleotide long RNA transcript. But the mature mRNA that is translated on the ribosome is 1872 nucleotide long. This observation suggests that the gene is okay. So if there are 7700 nucleotides, they are saying it is the mature. That means after the post-transcriptional modification, okay, it is having just 1872. Okay, 1872 nucleotides. That means what happens is after here there is a process called as splicing. In the process of splicing, what happens is the introns are removed. So after removing the introns, we have 1872 nucleotides. So when there is the existence of introns and exons simultaneously, this is called as the split gene. Okay, this is called as a split gene. So here the correct option is split gene. Next question is, agarose gel electrophoresis is routinely used for the analysis of DNA preparation. Which of the following cannot be achieved by the electrophoresis? Studying DNA protein interaction? No, we can study DNA protein interaction using this method. Resolving of DNA fragments of different lengths. So this is also possible. So this is wrong option. They are asking not achieved. Separation of different molecular conformations of DNA molecule. So yes, this is also possible. DNA sequencing is not possible using gel electrophoresis. So this is the correct option here. Next is, in most cases, immune cell that recognizes antigen in complex with MHC1 is. So here, the only cell is cytotoxic T cell. This is cytotoxic T cell. It contains MHC1 on its surface. Okay, it contains MHC1 on its surface. Macrophage, B cell, helper T cell do not contain the MHC1. Okay. Next, if the genotype of an individual is this, how many type of gametes are produced by this individual? Okay, friends. So we have to remember one thing. The formula for calculating the number of gametes is equals to 2 to the power n, where n is equals to number of heterozygous condition. Okay. So here let's see how many heterozygous condition are there. So here we see capital B, small b, capital C, small c. Here we have only two heterozygotes. So we write 2 to the power 2, which will be equal to 4. So the correct option is 4 here. Okay. Next is the genome size of M13 bacteriophage is. So the genome size of M13 bacteriophage, this is a conceptual question. It is 6.4 kilobase. Next question. In addition to tuberculosis, Robert Cochalso, known for the discovery of the causative organism for, so he is responsible for the discovery of cholera causing organism. Which of the following molecules is not a secondary metabolite? Here we see resveratrol, solasodine and anthocyanins. These three are secondary metabolites. Whereas malonic acid is not a secondary metabolite. So this is the correct option. Which one of the following is a sequence alignment tool? ProCyte, PIR and Print are not the sequence alignment tools. BLAST is a sequence alignment tool which is used to sequence the DNA. During one round of TCA cycle, how many molecules of carbon dioxide release? So there are 2 carbon dioxide which are released during the TCA cycle. Next, which of the following endonuclease is involved in the post-transcriptional gene silencing? So gene silencing is done with the use of dicer enzyme. Okay. You successfully cloned, it's not done, it's cloned, and express a gene encoding for an antigenic protein from SARS-CoV-2 virus, but when you injected this recombinant viral protein into mice to generate functional neutralizing antibodies, you failed to get any neutralizing antibodies. What would be the most probable reason? The most probable reason is that the purified protein was folded incorrectly. Okay, the purified viral protein is truncated. No, because truncated means shortening in the length. Why would it get shortened? So this is wrong. Mice had weak immune system. No, we would already use a mice which is having a good immune system. Mice do not respond to SARS-CoV antigen. There is nothing as such. Okay. Next question. Which of the following incorrectly describes a molecule with the mass of 1000 times that of water? So it is Mr 18,000 Dalton. DNA polymerase 1 has the following activities. 5' to 3' exonuclease, yes it has, so this is wrong, it is okay, so yes this has 5' to 3' polymerase, yes this activity also it has. 3 to 5' exonuclease, yes this activity also it is having. So all of the given options are correct. This third option is correct. Pyruvate dehydrogenase complex catalyzes the formation of acetyl-CoA from pyruvate. Which of the following describes this type of reaction that occurs? So here we can see pyruvate dehydrogenase is converting pyruvate to acetyl-CoA. So here in that process there is a removal of carbon dioxide. So this is called as oxidative decarboxylation. Okay, so here the correct option is oxidative decarboxylation. The average length of carbon-carbon single bond is 0.154 nanometer. The enzymes involved in the electron transport system are mainly located on the, okay, see, this is the outer mitochondrial membrane. Okay, and here you see this is the inner mitochondrial membrane. So on the inner mitochondrial membrane, this is the electron transport system which is present. So here the correct option is on the inner mitochondrial membrane. Okay, next, nitrogen fixation is. It is a conversion of nitrogen to ammonia. It is called as nitrogen fixation. CRISPR-Cas9 system can be used for. It is used for editing the nucleic acid. So this is the correct option. Why is mitochondrial DNA considered to be the best marker for phylogenetic analysis? So what happens is, suppose this is the egg and this is a sperm. My pen is not working here. Okay, so from the sperm we derive nucleus, but from the egg we derive nucleus plus mitochondria. Okay, so that is the reason why mitochondrial DNA considered to be the best marker for phylogenetic analysis, because mitochondrial DNA shows a maternal inheritance. It is inherited from our mother. Next, addition of salt to a culture medium allows only the salt-tolerant bacteria to grow. This is an example of selective media. So what is selective media? Selective media, what it does is, suppose I want to grow only the salt-tolerant bacteria. Okay, so in the selective media, I will add salt. So it will allow only salt-tolerant will grow. All others will die. So it is only allowing the growth of salt tolerant bacteria. So it is a selective media. Structural alignment is measured by, it is measured by RMSD. RMSD means relative mean standard deviation. Okay. Yes. Next, low expression of protein is observed in given tissue. Which of the following techniques will be used to determine whether defect is at the transcriptional level or translational level? So study the the protein expression at the transcriptional level. We use northern blotting. Okay, we use northern blotting. And to study at the translational level, we use western blotting. And if we want to see from DNA to RNA, if we want to study the RNA, then we use southern blotting. Here they are asking transcriptional and translational. So northern blot and western blot is the correct option here. Okay. So hope you guys have understood. Thank you, friends.